Everyone suffers from stress, whether it be from the dangers of encountering a bear or from studying for a major biology exam. Increased heart rate, sweat forming on your brow, clammy hands, insomnia, stomachache, indigestion, and that overwhelming feeling of despair are all just too familiar. We've all heard of the saying that we should always follow our gut instincts, but what if we told you that there might be a way to combat the effects of stress by using our guts?
As you know already from our class's study of the digestive system, microbes play a crucial role in nutrition, digestion, and absorption. However, their roles may be more extensive than that. Researchers at the University of Colorado at Boulder hypothesized that prebiotics-rich diets reduce the disruptions of the diurnal cycle caused by severe or chronic stress by supporting gut bacteria populations. By studying the effects of a test diet rich in prebiotics on lab rats over the course of several months, the researchers showed that a diet rich in prebiotics started early in life can promote phenotypes in the gut microbiota that includes protection of the diurnal cycle and moderation of sleep patterns.
So how exactly did these researchers go about their research? Well, it turns out that their experiment called for rats, 54 of them, all males. Their choice and strain of rats lies in the fact that they respond consistently to stressors, in addition to the fact that this allows them to minimize variability by using the same kind of rats, all only 24 days old. The reason why such young rats were chosen is because of how malleable gut bacteria are at early stages of life. By malleable, we mean that external factors like exercise and a diet of prebiotics can have an effect on the bacteria to make them more likely to buffer the effects of stress in adulthood. When the rats were 59 days old, biotelemetry transmitters were surgically implanted into their intestines in order to measure the core body temperatures. Electromyography leads were then put into the back of their necks in order to record how active their skeletal muscles are, followed by EEG recording electrodes into the base of their skulls to measure sleep cycle. When the rats were around 87 days old, half of the rats were exposed to 100 tail shocks in a separate room. After shock exposure, the rats were returned to their cages where the body temperatures were recorded for the next 6 days. Fecal cultures were also looked at in the control and test diet rats in order to determine whether or not there was an increase in bacterial species in the gut due to prebiotics.
Data were examined and analyzed using a ANOVA test. Post-hoc analysis is also used when appropriate using Fisher's PLSD with the alpha value set to p's less than 0.05. All the conclusions have statistically significant results. Figure D shows that stress flattened the diurnal amplitude of core body temperature in both the control diet group and the test diet group. However, the test diet group had a significantly less decrease in core body temperature compared to the control diet group. This demonstrates the test diet's ability to attenuate the effect of stress on diurnal amplitude of core body temperature. This low level of Deferibacterius correlates with longer NREM episode durations at PND72 as shown by the solid black line on figure A. Test diet also increased the level of proteobacteria. Higher level of proteobacteria correlates with longer REM sleep in rats after stress.
The results of the experiment clearly support the hypothesis that a diet rich in certain prebiotics reduces the effect of acute stressors on REM sleep and mitigates disruptions in the day-night cycle. The rats that consumed the test diet exhibited higher levels of REM sleep after exposure to electric shock stressors and were better able to regulate their temperature cycle throughout the day and night. The test rats clearly were able to handle stress better than their control counterparts, but the presence of prebiotics was not the direct cause of the newfound hardiness. The study looks deeper and shows that the improvements in stress responses correspond to the diversity and abundance of gut microbacteria. While these results do not directly apply to humans, it does suggest that a solid link may exist between our stomach bacteria and our overall health as an organism.
So how does stressing rats out relate to us? Well, there is no way for us to conclude with the results from this experiment that the same results can be replicated in any other species. It is a known fact that mammals rely on symbiotic relationships with bacteria for essential functions, just like the rats in the experiment. What's more, since humans and rats are both mammalian omnivores, the structures of digestive tracts of both species are more similar than they are different, with the main difference being that humans require more compartmentalization and specialization in their organs. More differences do emerge when looking at the microbiome of the two species, however. While mice and human microbiota are dominated by bacteria of these two same major phyla, one study shows that 85% of the bacterial genera found in mice are not present in the human gut, meaning that the results from this experiment probably can't be expected to be the same in humans. So even though humans and mice might not share the same exact kind of bacteria all throughout their digestive tracts, there is no doubt that the shared major role of microorganisms in mice and human guts is to help digest food and absorb nutrients. And though we can't exactly just directly apply these findings to humans, this is the first experiment to ever show with concrete evidence that a prebiotic diet can provide support to fighting stress on sleep-wake behavior. If this specific diet could have such a major effect on the response of rats to stress, maybe this could be the next step in fighting stress in humans.
Created using Powtoon.